Welcome back to another episode of the College of Glycation, where we look into and discuss the hidden science of how sugar's dark side shapes our health. Once again, my name is Paul Reynolds. I'm a biomedical scientist and professor of cell biology, and I'm also going to be your host for today's episode. If you've been following along, you know that we've spent time in each of the previous episodes laying the groundwork on glycation. That's the sneaky process where sugars, like glucose, bind to proteins, lipids, and even your nucleic acids in cells throughout the body. In the process, they form advanced glycation end products, or AGEs. And these AGEs aren't harmless byproducts. They're like molecular graffiti that stiffens tissues and sparks inflammation, eventually accelerating aging at the cellular level. Think of it as sugar's way of leaving permanent stains on the machinery of your life. Today, we're turning our lens to the liver, that unsung hero that filters toxins and metabolizes everything we throw at it, or tries to. Our episode is called Glycation and Liver Disease, from Fatty Liver to Cirrhosis. In this episode, we'll explore how glycation fuels a silent epidemic that's exploding worldwide, non-alcoholic fatty liver disease, or NAFLD, and its aggressive cousin, NASH, often flying under the radar until it's too late. By the end, you'll see how catching it early matters and how simple shifts in diet, movement, and even stress management can turn the tide. So let's dive in. First, a quick refresher for those tuning in mid-series. Glycation kicks off when blood sugar levels run high, or as in the case of insulin resistance, where your bodies do not respond well to insulin. It's also common in unchecked carb-heavy diets. Excess glucose doesn't just float around. It actually latches onto proteins like collagen in your skin, in your blood vessels, and in many other cells throughout the body, creating these rigid AGEs in the process, known to trigger oxidative stress. That's where free radicals run around causing problems, as well as inflammation. In this case, the body's misguided alarm system. We've talked about how this process shows up in stiff arteries or foggy brains, but other areas as well. But in the liver, that's ground zero, and that's our focus today. This organ, the liver, processes 70% of the nutrients that you receive from your gut, turning carbs into energy or fat, and fats into fuel. When glycation enters the mix, however, it doesn't just tag along. In fact, it hijacks the controls, leading to extra fat buildup, scarring, and eventual organ failure. Here's the kicker. Non-alcoholic fatty liver disease, or NAFLD, affects 1 in 3 adults globally right now, up from obscurity just a few decades ago. And it's driven by our love affair with processed foods and sedentary screens. It's often asymptomatic. There's no pain, there's no warning, until fibrosis turns into cirrhosis, a portal of hypertension or worse. But knowledge is power, so let's break down the mechanisms backed by solid science and arm you with actionable steps. So let's start with the basics of how insulin resistance works in this scenario. Insulin is a hormone made by the pancreas, and its most famous job is to lessen blood glucose levels by causing cells to absorb the glucose. In this case, insulin resistance is the inability of your cells to respond properly to insulin, and that drives fat and sugar straight into the liver's lap. Insulin is the key that unlocks cells to absorb glucose for energy. So when insulin resistance hits, glucose piles up in the blood and in the liver. Sensing chaos, it ramps up fat production from scratch, a process called de novo lipogenesis. Meanwhile, free fatty acids form a struggling adipose tissue flood, and this causes extra fat buildup. Fructose from sugary sodas bypass regulation, heading straight to the liver for conversion to triglycerides. The result, hepatic steatosis, or fatty liver, where up to 50% of the organ's weight can become fat. And this process, I promise, is not benign. It is the spark for non-alcoholic fatty liver disease. Studies, in fact, show that insulin resistance is the hallmark here, linking it tightly to metabolic syndrome, which includes high blood pressure, belly fat, and dyslipidemia, or problems with lipid levels. In one comprehensive review, researchers detailed how this insulin resistance reduces the liver's ability to oxidize fats. In the process, it traps them inside the liver like junk in a hoarder's attic, while excess sugar fuels glycation, making the mess even stickier. It's a vicious cycle. Fat impairs insulin signaling further, sugar accumulates more, and glycation amplifies the damage by cross-linking proteins, impairing liver enzymes, and promoting inflammation. So let's now zoom in on glycation's dirty work in the liver specifically, starting with hepatic fibrosis, the scarring that bridges fatty liver, accumulation of fat in the liver, to cirrhosis itself. Fibrosis happens when the liver's repair mechanisms go haywire, laying down excess collagen like overzealous builders patching a leaky roof with steel beams. It's not appropriate to have that much collagen. Central to this are hepatic stellate cells. Those are quiet or quiescent guardians in healthy livers that store vitamin A. But under stress from all the ages that accrue, these cells awaken and they transform, instead of quiet supportive cells, into myofibroblasts. And their job is to churn out extracellular matrix like collagens and the fibrous scaffolding that stiffens your tissue. Glycation accelerates this by binding to receptors on stellate cells, generating reactive oxygen species in the process. Those are small rogue molecules that damage cells. And they also, by way of this binding, upregulate genes for alpha smooth muscle actin and collagen type 1. The more muscle you have and collagen, the more fibrosis you'll become. In lab models, exposing stellate cells simply to ages boosted their proliferation and activation, directly linking high sugar environments to fibrosis progression. Imagine your liver's support cells turning from librarians to construction crews overnight. They were once housekeepers, but now they are building collagen abundantly. All of this because sugar's graffiti signals emergency remodel in the process. Without intervention, this fibrosis advances silently, compressing blood vessels and leading to the nodular, shrunken liver we see in those that have cirrhosis. So let's enter into the story this idea of RAGE, that is the receptors for ages. It is a cell surface sentinel that when it binds these glycated ages, it unleashes a cascade of trouble in non-alcoholic fatty liver disease progression. RAGE isn't picky. It's not selective. It grabs any ages that are present and kicks off signaling pathways that are inflammatory. One of the pathways that is most central to its effect is that is governed by NF-kappa B, which dials up pro-inflammatory cytokines such as TNF-alpha and IL-6, ramping up this inflammatory environment. In the liver, this means that comfort cells, those are the macrophages that float around with the job in the liver of cleaning debris, they get riled up, they get cranky, and they recruit even more immune players in the process, amplifying oxidative stress. From there, it's a straight shot to stellate cell activation and fibrosis. Now, there have been many studies that support this. Animal studies in which animals are fed high fructose, high cholesterol diets laced with ages showed worsened steatosis, ballooning hepatocytes, massive fat and fibrotic hepatocytes, and collagen deposition. Now, all of those outcomes, which are problems, were also shown in animal models to be decreased or dialed back when age-rage signaling is blocked. Another model using a methionine choline deficient diet that mimics non-alcoholic fatty liver disease found that ramping up dietary glycotoxins via rage drove liver age accumulation, oxidative bursts, and profibrotic shifts in stellate cells. All of these outcomes push the disease from fat blobs to inflamed, irreversible scars. Now, rage signaling isn't just a side player, it is the amplifier that turns reversible fat into irreversible damage. And that explains why non-alcoholic fatty liver disease and other such liver anomalies, rates of those effects are skyrocketing in sugar-saturated societies like ours. This brings us up to the explosion of these diseases worldwide, a crisis often overlooked until late stages. In the US alone, it's the top reason for liver transplants. Yet most folks learn of it incidentally via blood work or imaging. Globally, prevalence has quadrupled since 1980, hitting 38% in some populations, fueled once again by ultra-processed foods high in hidden sugars and fats that spike glycation. So what are the symptoms? Well, fatigue, vague right upper quadrant discomfort, that's where your liver mostly is found, maybe elevated ALT or AST enzymes, dismissed as nothing serious. Those can be the early harbingers of these outcomes. By the time ascites or jaundice appears, fibrosis has progressed most likely to cirrhosis, with risks of liver cancer short behind. The tragedy is its preventability. Early detection via fibroscan or even simple ultrasound paired with lifestyle tweaks can halt the progress. But ignorance persists. Doctors screen sporadically. Patients ignore the metabolic red flags. It's like driving with a slow tire leak. It's fine for a while until you're eventually stranded. So how do we fight back? Well, let's talk about some of the strategies that can be helpful. All of these are grounded in evidence, and we're going to focus on our ability to slash glycation and inflammation. We can do that through diet, exercise, and stress management. Diet is king here. Low-carb approaches, limiting sugars and refined grains to under 50 grams daily, will curb de novo lipogenesis, new fat formation, as well as insulin spikes. In this case, we'll starve the liver of excess fuel for fat and ages. Ketogenic variants high in healthy fats like avocados or olive oil can also flip the metabolic switch to fat burning, reducing hepatic triglycerides by 30 to 50% in some clinical trials. Let's talk about intermittent fasting. Let's say you eat within an 8 to 10 hour window. This mimics caloric restriction, boosting autophagy to clear damaged cells and glycated debris. And it also, I promise, will improve your insulin sensitivity. There was a randomized trial that pitted a 5-to-2 fasting regimen, that's 2 low-calorie days weekly, against a low-carb, high-fat, and standard care in non-alcoholic fatty liver disease patients. Both of those outperformed controls. In fact, it dropped steatosis or fatty liver by 7 to 8% as revealed by MRI, with fasting edging out on insulin resistance metrics. Meta-analyses confirm these outcomes. These patterns not only shrink liver fat, but they also lower ALT and inflammation markers, all without the yo-yo of the crash diets. Here's a pro tip. Start with time-restricted eating around your natural rhythm. Dawn until dusk, for most, is easiest. You're sleeping usually. And you can pair that with anti-glycation foods like berries, green vegetables, and spices such as cinnamon, which inhibit age formation. I'd like to also advocate that exercise is not optional. It's the accelerant in the process of healing. Now, I understand that many in the audience may struggle with different types of exercise. The goal here is simply move. Anything you're able to do effectively is better than not doing anything at all. Of course, aerobic exercise like brisk walking or cycling for 150 minutes or so weekly can help because it ramps up mitochondrial function, oxidizing fats, and quelling oxidative stress from glycation. Resistance training is also very helpful. It builds muscle, which is a sink for glucose that eases insulin resistance. The more you're able to build or maintain muscle over time, the more glucose can be pulled out of the blood, ameliorating glycation. Now, in MAFLD, that's metabolic fatty liver disease. That's the updated term for metabolic fatty liver. A position statement from exercise physiologist recommends combined aerobic and resistance protocols. It shows that 20 to 30% steatosis reductions or fatty liver reductions can occur independent of weight loss. So the scale may not show you're losing weight, but you're losing liver fat, which is helpful. All of that occurs via lowered RAGE expression and decreased inflammation. Think of it as hiring a cleanup crew for your liver's overworked factory when you combine these types of exercise routines. Now, let's not sleep on stress management. I alluded to that at the start of our episode. Chronic cortisol from work deadlines or endless scrolling spikes sugar in the blood, feeding glycation and inflammation. So there are many techniques like mindfulness, meditation, or yoga that have been clearly shown in the literature to lower reactive oxygen species, decrease inflammatory NF-kappa B activity. All of those are key in the RAGE pathway. While direct liver trials are emerging, broader evidence ties stress reduction to better antioxidant defenses, curbing progression in oxidative-driven diseases like non-alcoholic fatty liver. One study in particular on dietary interventions for oxidative stress highlighted how caloric patterns that include stress-lowering mindfulness amplify the benefits. In those cases, reducing inflammatory cytokines by a full 25%. So let's integrate it. A post-dinner walk with deep breaths or 5 minutes of box breathing before bed can be very helpful in addressing stress levels. So let's wrap up. Glycation isn't your destiny. It's rather a signal to reclaim control. From insulin-driven fat foods to RAGE-fueled fibrosis, the path from non-alcoholic fatty liver disease to cirrhosis is paved with excess sugar. But you can detour from that road with your deliberate choices. Monitor your markers, tweak your plate, move your body, and quiet your mind. Your liver will thank you. Not with grand fanfare, most likely, but with quiet, healthy resilience. Thanks everybody for joining me in this episode of the College of Glycation. When we understand how glycation damages tissues, we can take steps to interrupt that process and rebuild. Thanks again for listening, everyone. Until next time, stay curious and stay metabolically healthy.